Hello everyone. This section is for bacterial cell morphology. After learning this section, you should distinguish a typical bacterial cell from a typical plant or animal cell in terms of cell shapes and arrangement, size, and cell structures, and discuss the factors that determine the size and shape of a bacterial cell. The microbial world offers considerable variety in terms of morphology. However, the two most common shapes are cocci and rods. Cocci are roughly spherical cells. They can exist singly or can be associated in characteristic arrangements that can be useful in their identification. Diplococci arise when cocci divide and remain together to form pairs. And you can see long chains of cocci result when cells adhere after repeated divisions in one plane. This pattern is seen in the general Streptococcus, Enterococcus, and Lactococcus. Members of the genus Staphylococcus divide in radium planes to generate irregular, grape-like clumps. Divisions in 2 or 3 planes can produce symmetrical clusters of cocci. Look at the picture. Bacteria in the genus Micrococcus often divide in 2 planes to form square groups of 4 cells called tetrads. In the genus Cecina, cocci divide in 3 planes, producing cubical packets of 8 cells. Bacillus bacterium is an example of a bacteria with a rod shape. See the figure. Rods, sometimes called bacilli, differ considerably in their length to width ratio, the cocobacilli being so short and wide that they resemble cocci. The shape of the rod's end often varies between species and may be flat, rounded, cigar-shaped, or bifurcated. Although many rods occur singly, some remain together after division to form pairs or chains. For example, Bacillus bacterium is found in long chains. Other cell shapes and arrangements are also observed. Vibrios are comma-shaped. Spirilla are rigid, spiral-shaped cells. Many have tufts of flagella at one or both ends. Spirochetes are flexible, spiral-shaped bacteria that have a unique internal flagellar arrangement. You can see it on this picture. These bacteria are distinctive in other ways, and all belong to a single phylum, Spirochetes. The oval or pear-shaped members of the genus Hyphomicrobia produce a bud at the end of long hypha. Other bacteria are pleomorphic, being variable in shape and lacking a single characteristic form, such as star-shaped bacteria, square-shaped bacteria, prosecate bacteria. Some bacteria can be thought of as multicellular. Actinomyces typically form long filaments called hyphae. The hyphae may branch to produce a network called a mycelium. And in this sense, they are similar to eukaryotic filamentous fungi. See the figure E. It is a SEM micrograph showing a kind of filamentous bacteria. Many cyanobacteria, a group of photosynthetic bacteria, are also filamentous. Being filamentous allows some degree of differentiation among cells in the filament. For instance, some filamentous cyanobacteria form heterocytes within the filaments. These are specialized cells that carry out nitrogen fixation. The mixer bacteria are of particular note. These bacteria sometimes aggregate to form complex structures called fruiting bodies. You can see the picture F. Size. Then let's talk about the size of bacteria. Escherichia coli is an excellent representative of the average size of bacteria. This rod-shaped bacteria is 1.1 to 1.5 micrometer wide by 2 to 6 micrometer long. However, the size range of bacterial cells extends far beyond this average. Near the small end of the size continuum are members of the genus Mycoplasma. It's about 0.3 micrometer in diameter. At the other end of the continuum are bacteria such as some spirochetes, which can reach 500 micrometer in length, and the cyanobacterium oscillatoria, which are about 7 micrometer in diameter, is the same diameter as a red blood cell. The huge bacteria, Impelopasium facialsoni, lives in the intestine of the brown surgeonfish. Impelopasium facialsoni grows as large as 600 by 80 micrometer, a little smaller than a printed hyphen and clearly larger than the well-known eukaryote paramecium. You can see it on the picture. An even larger bacterium, Thymogenetor namibidinus, has been discovered in ocean sediment. You can see it on this picture. There is a doubt. The variety of sizes and shapes exhibited by bacteria raises a fundamental question. What causes a bacterial species to have a particular size and shape? Although far from being answered, recent discoveries have fueled a renewed interest in this question, and it is clear that size and shape determination are related and have been selected for during the evolutionary history of each bacterial species. For many years, it was thought that microbes had to be small because being small increases the surface area to volume ratio. As this ratio increases, the uptake of nutrients and diffusion of these and other molecules within the cell become more efficient, which in turn facilitates a rapid growth rate. Shape affects the SV ratio. A rod with the same volume as the coccus has a higher SV ratio than does the coccus. This means that a rod can have greater nutrient flux across its plasma membrane. However, the discovery of Impulopicium physiolosoni demonstrates that bacteria can be very large. For bacteria to be large, they must have other characteristics that maximize their SV ratio, or their size must be beneficial in some way. For instance, this bacteria has a highly convoluted plasma membrane, which increases its SV ratio. In addition, large cells are less likely to be eaten by predatory protists. Cells that are filamentous, have porosity, or are oddly shaped are less susceptible to predation. That's all for this section. Here is a word list for you.